Hi everyone, Brenda Gustin here, and we're on the last part of chapter 3. We're going to be talking about the internal structure of a bacterial cell. And um, remember that we started off talking about things on the very outside of the cell. We talked about the flagella and extracellular polysaccharides, the capsule and the slime layer. And then we got onto the cell wall where we talked about what makes a cell gram-positive, gram-negative, or acid fast. And then we got to plasma membrane, the cytoplasmic membrane, which is deep to the cell wall. So now we're inside of that cytoplasmic membrane, and we're going to talk about some parts. Before we do that, I'm going to go back to section 3.1. So I'm going to just back up right here and remind you of this picture. I'm going to get my face out of the way. There we go. And so this is a great place to go back and remind yourself. And so the parts that we're going to talk about inside of the cell today, we're just going to mention, so inside of the cytoplasmic membrane, where is it? Right there, that yellow layer. So now we get in there, we're going to talk about the bacterial chromosome or nucleoid. And so you can see this big kind of folded up piece of purple right there. Ribosomes, there's, I don't know, I think hundreds, thousands of ribosomes inside a bacterial cell, all these little dots. And you might remember they're the site of protein synthesis. We're going to talk about each one of these separately. The cytoplasm, the water-based interior of the cell. Let's see, what else are we going to talk about? I'm going to mention the concept of inclusions. And inclusions are just chemical deposits in a cell, and I'll mention that. Plasmids. Plasmids are actually little pieces of DNA. So what you know about DNA, the double helix, the plasmids are also little pieces of DNA, and I'll try to help you understand why that's important. And then finally, the last thing we're going to talk about are endospores. So let's go back to section 3.4. There we go. So here it talks about the cytoplasm, and it's a gelatinous solution inside the cytoplasmic membrane, mostly water, and it's just got a lot of stuff in there that's important for cells to function. Complex mixture of nutrients, sugars, amino acids, and salts. So that's the same thing as is in eukaryotic cytoplasm. So that's a kind of a typical thing for cells. When we look at the bacterial chromosomes, now hopefully you remember from previous classes that your chromosome is made of DNA. And if you think about human chromosomes, see if you could, what you remember about that. Let's see, in the human cells, in eukaryotic cells, the chromosomes are inside of what organelle? The nucleus, but remember prokaryotes don't have a nucleus. The other thing is if you think about human chromosomes, you might remember that they're linear pieces of DNA. And see if you remember how many chromosomes are in each one of our cells. Hopefully you came up with either 46 chromosomes or 23 pairs of chromosomes. Well, that's a lot different than a bacterial cell. Bacteria have many fewer functions compared to many eukaryotic cells, and turns out the bacterial chromosome is typically a single circular piece of DNA. Let's see, it's a long piece of DNA. And it says long helix formed into a circle. So in a bacterial cell, instead of having many linear pieces of DNA, it actually is just one circular piece of DNA in a bacterial cell. Now, when you looked at that picture on section 3.1, it kind of looked like it was all tangled. And so what it is you take this circular piece of DNA, but it has to be kind of folded up and squished up to fit inside of that cell. So that's why it doesn't necessarily look like a circle when you're looking at that diagram. And hopefully you remember a little bit about DNA, how it's divided into units called genes, and the genes encode for different proteins to provide different functions for the cell. There actually, in many bacterial cells, there are other little pieces of DNA called plasmids, and you could see that in the diagram in section 3.1. And plasmids are just very small pieces of DNA compared to bacterial chromosomes. But the plasmids, again, are just like the bacterial chromosomes. They're circular pieces of DNA. But the genes that are on plasmids are not necessarily essential for normal life in the bacterial cell. So I always think about plasmids as being pieces of DNA that give the bacterial cell a superpower. And so here they're saying these plasmids are, when the cells are reproduced, they're passed on to the offspring, but the genes there are not essential to bacterial growth and metabolism. But they do provide protective traits such as resisting drugs, producing toxins and enzymes. And so one of the big reasons why we talk about plasmids in this course is because it's a common way for one bacterial cell to transfer antibiotic resistance to another cell. And we'll talk about that more when we get to talk more about genetics. But plasmids give the bacterial cell a superpower. So if you get a plasmid that has a gene that helps you resist penicillin, you will become penicillin resistant. And that's a pretty big deal. The one other part of the inside of the cell are ribosomes. And again, before you even took this class, hopefully you remember that eukaryotic ribosomes are the site of protein synthesis. They're where proteins are made. And it's the same function as in bacterial ribosomes. So bacterial ribosomes and eukaryotic ribosomes are both where the RNA hooks up and makes proteins, the site of protein synthesis. Again, we'll talk more about that as we get to genetics, but it's really important. It says you can have a bacterial cell can contain thousands of ribosomes. So making proteins is absolutely essential to the life of a cell. It goes into a lot of detail about the structure of ribosomes, but really one of the more interesting things about them is that eukaryotic ribosomes are similar, but a little bit different in their structure. And because of that, eukaryotic ribosomes and prokaryotic ribosomes kind of respond differently to certain drugs. Specifically, we're talking about some antibiotics like streptomycin and tetracycline that kill bacteria but don't kill our own cells. So because the ribosomes in bacteria are slightly different than those in eukaryotic cells, it's a great target for a drug. You can kill the bacteria without killing the eukaryotic cell. If you stop ribosomes from working, the cell's not going to survive. See if you can remember what the other target we had for drugs in bacterial cells. Remember, it has to be something that's different in bacteria than the host. Hopefully you remember that it's the bacterial cell wall, the peptidoglycan, that's unique to bacteria. We don't have it in eukaryotic cells. So again, if you can find an antibiotic that sticks to it, and penicillin happens to be one, you can kill the bacteria without killing the host. This word inclusion, another part that I wanted to talk about, I generally talk about it because in live classes sometimes, or even in pictures, you're looking at pictures of bacteria and they look like they have organelles in them. They look like there's something inside of them. And in this one, they kind of highlighted the inclusions, but these would be like dark dots in there. You remember we said bacteria don't have membrane-bound organelles, so it can be confusing. Like, what are those things inside the bacterial cells? And I would generally say, oh, there's some kind of inclusion, just to sound smart. But an inclusion just means some kind of nutrient that's being stored. So it says they can store nutrients so they can respond to periods of low food availability. So some bacteria will store certain things inside of their cells to use for later. They also said some inclusions are bacteria put gas in these little structures and it helps them float to the surface. It gives them buoyancy. So there's all different kinds of reasons why bacteria might have inclusion. So if you see a picture of bacteria and they look like they have something inside them, like organelles, you can just say, oh, they must be some kind of storage product, some kind of inclusion. You know, I just realized I forgot to mention one vocabulary word. When I went, if we were looking at DNA and looking at the chromosome, they gave you this word nucleoid. Remember that bacterial cells don't have a nucleus. They don't have a membrane-bound nucleus. But if you look in pictures, like on electron microscopes, there's a dark area in there that looks like it's, it might be a nucleus. What is that? Well, it's the nuclear region. It's where the chromosome is all kind of bunched up in there. So you might see what we call a nucleoid or a nuclear region in a picture of a bacterial cell. The last thing I want to talk about are endospores. And meanwhile, rather than me do it here, you can go in the book and do these NCLEX preps. They're really good, and they give you little answers here that you can practice. You can't get too much practice on NCLEX prep if you're going into nursing. Okay, so bacterial endospores, these are something that only some bacteria make. And in fact, these would be pretty special bacteria. It says they're dormant bodies produced by bacteria such as Bacillus and Clostridium. I'm not going to mention this last one, but I would like you to remember these two genera. Bacillus, remember Bacillus is a shape, but it's also a genus. And another genus called Clostridium. Now, I'm going to give you an exception to that there's one kind of bacteria that used to be called Clostridium, but now is Clostridioides. But you can just write C and recognize that. I'll show you that in a minute. But endospores are dormant bodies produced by bacteria in the genus Bacillus and Clostridium. And the bacteria can exist in two forms, a vegetative cell and an endospore. So vegetative cells are bacteria that are doing their normal thing. They're growing, they're metabolically active. Being a vegetative cell for a bacterium is a normal thing to do every day. It's just actively growing. But bacteria that can make endospores, sometimes environmental conditions become challenging. And here they're saying what kind of things can happen. I think they tell you down here. They can survive extreme heat, drying, freezing, radiation, and certain chemicals. So let's say you've got bacteria that are kind of sitting in the soil and it's starting to get, we haven't gotten rain for a few days. It's starting to dry out or it's getting very warm. And it might trigger these vegetative bacterial cells to sporulate or go through sporulation and make an endospore. So you can think of the endospore kind of like a time capsule around the DNA in the bacterial cell. So this endospore is formed inside the bacterial cell, and all it's doing is saving the DNA. The bacterial cell is no longer metabolically active, and it can just sit there. Now, how long can an endospore just sit there and the bacteria do nothing? It can be days, weeks, months, even here we have one right here. It says microbiologists have unearthed a viable endospore from a 250-year-old salt crystal, 250 million-year-old salt crystal. So some of these endospores can last a very long time. They're extremely stable and can just sit there and wait for conditions to get better before they become bacteria again. So let's look at the process of sporulation and another step we call germination. This picture is good. What it's showing you, and I'm just going to go over the real basics of it, but here's a vegetative cell, a bacterial cell with all the right parts, and there's the chromosome there, that circular chromosome that's all puckered up, all folded up. But something's going on that things aren't going well. So it's getting too hot, the cells are running out of nutrients, and this has to happen over a period of time, over several hours. And so what the cell does is it copies its DNA and it starts forming some layers around one part of the DNA. So this process right here, we're going to call sporulation, where the endospore is forming. So down here, we've got a point, we had the vegetative cell, but now it's got an endospore inside of it. And eventually what happens is the vegetative cell just kind of dies off, it disintegrates, and you just have an endospore. And this thing, like we said, can sit there for many, many years until, and it might just be few hours. It might be years to hours. It doesn't matter. It just depends on what the situation is. But if things start getting better, like, oh, it rained and now there's moisture again, those endospores can germinate and become vegetative cells again and do the normal cell division like bacteria would. So the process of sporulation is when bacteria in the genus Bacillus or Clostridium are under stress, and so they're going to make endospores. And then the endospores will eventually just survive through the bad times. And then once they get exposed to better conditions, they're going to they're going to germinate and turn back into a vegetative cell, an actively growing cell. Now, this has a really big importance for disease. And what is the medical significance? Well, it says the majority of endospore-forming bacteria are relatively harmless, but there's some that are really important that cause pretty big diseases. And one is caused by Bacillus anthracis, causes the disease anthrax. And you might have heard of anthrax because it can cause disease in animals, in like cattle, and animals related to cattle. It can cause disease in humans, and it can be deadly. If you inhale it, it's a very deadly disease. And it's been used for bioterrorism. So why could it be used for bioterrorism? Because it makes endospores. If you ship some E. coli through the mail, it would just pretty much die because it doesn't have enough moisture in food. But if you ship some Bacillus anthracis that's making endospores, those endospores, they don't need water, they don't need food. They can sit there and wait, and you open an envelope, which has been done with weapons grade anthrax in it, and people inhale it, and it can cause death or very serious disease. Remember, the other genus of bacteria that make endospores is Clostridium. And you've heard of, we all get our tetanus shots. And so tetanus is, you might have heard it called lockjaw. You get, your muscles get, they stay contracted. So you can get your lockjaw is one of the early signs where your jaw can't move. But Clostridium tetani is from a bacteria that makes endospores. Well, a lot of these endospore forming bacteria are in soil. So that's why you hear about, oh, if there's a rusty nail. Well, it's an old nail that it's been outside, so it's rusty, might have some dirt on it, might have some Clostridium tetani endospores in it. And so that could cause somebody to get tetanus. You may or may not have heard of gangrene. When somebody has a tissue injury, like a injury to their body where oxygen isn't getting into the tissue, and there could be dirt exposure. I mean, there's bacteria, these endospore formers can be in dust because they don't need nutrients to survive. They can just be in the form of endospores. So if you get exposed to Clostridium perfringens, which is just around, you could get gangrene, and you might have seen pictures of tissue turning black and people having to get amputations because of it. Clostridium botulinum causes botulism, which is the worst food poisoning you can get. And it's funny because it's related to Clostridium tetani, but Clostridium tetani causes your muscles to contract and stay that way, whereas Clostridium botulinum causes your muscles to relax and stay that way, which could again be deadly. So why do we have to, when we can food, why do we have to heat it above boiling? Why do we need pressure? To kill the endospores. Endospores, because they're so resistant, do not easily die in the presence of boiling water. Vegetative cells will die if you boil water, but endospores will not. So we need, it's talking about down here that ordinary boiling will not destroy endospores, so canning food must be pressurized. It talks a little bit about here how endospores inhabit soil, dust, and so cleanliness is important. And one of the big diseases that happens in hospitals frequently is one that you hear called C. diff. And this is the one that they changed the genus from Clostridium to Clostridioides. But we still start it with C. So C. diff or Clostridioides difficile. And that also is an endospore former. And I always, in my microbiology classes, I always have people from nursing that have had patients with this. This is really bad diarrhea. It happens a lot of times in older patients, and we'll talk more about it this semester. But it's important for you to remember that this disease is caused by a bacterium that can make endospores, because now some of that, diarrhea kind of gets everywhere. Well, if it's not, the room isn't cleaned adequately, and there's little droplets of feces around, you could have C. diff endospores there, and they could remain there for years. And so other patients could be exposed to them. So cleaning hospital rooms is really essential, and doing it correctly is really critical when dealing with diseases like Clostridium difficile. At that point, I'm going to call it the end.